Dr. Barr, and I love me some histology. Today we're going to talk about epithelial tissues and microvilli. From this section, I want you to be able to describe the ultrastructure of microvilli and correlate with their function and location. So in these next few lectures, we're going to talk about apical domain structures, which are very unique to epithelial cells. These are termed as surface modifications because the plasma membrane is altered in these types of cells. The structures include microvilli, also known as brush borders, stereocilia, as well as cilia. And in this particular lecture, we're going to focus on microvilli and stereocilia. Cilia will be discussed in a separate lecture. So what's the difference between villi and microvilli? Villi, by definition, are finger-like projections. Typically, we see villi in the small intestine, which are lined by simple columnar epithelium. So if you have this image here, this kind of shaggy rug-like structure are the villi of the small intestinal tract. Now, when we take a look at it ultrastructurally, the microvilli are finger-like extensions of the cell membrane. So it's basically you have villi on top of villi, right? Cool, huh? So microvilli are the ultrastructure finger-like extensions of the cell membrane. Villi is the overall organization and arrangement of the simple columnar epithelium, particularly in the small intestinal tract. Now, why do we need villi upon villi? Why do we need microvilli? And the function is mainly to increase surface area and maximize absorption, right? That's the role of the small intestinal tract. All the nutrients coming in have to be absorbed. And, the transit time can be fast, so we need to kind of maximize absorption and increase the surface area. Again, we talked about small intestinal tract, but you can also see microvilli in kidney tubules. Recall again from the first lecture of histologic features, I mentioned the proximal renal tubules have reabsorption properties or functions. And so that is microvilli are important for maximizing the reabsorption function in that location. So let's talk now about the microanatomy of an epithelial cell. How do we determine cell orientation and polarity? And this is determined by the distribution of membrane proteins, lipids, and the arrangement of the cell cytoskeleton. There are 3 geometric domains, and so in this case here you have the apical, which is basically the top side or the head side here. That's the region that is exposed to the lumen or the internal environment. Then you have your bottom or your basal area of the cell, which separates the cell from the underlying connective tissue attached to the basement membrane here. On the lateral surfaces there you have the cells that are adjacent to each other, right? The end to end, the side to side is your lateral side. You may come across the term basolateral, which is basically this U-shaped area here, the bottom and the lateral aspects. So each cell here is bound to each other via tight junctions, which we will talk about in a separate lecture. Now let's take a look at the ultrastructure of microvilli, and the key structure in the microvilli are these cross-linked actin microfilaments. That is the cytoskeletal structure of the microvilli. You can see in the image here you have your cross-linked actin microfilaments that form the bulk of the microvilli. At one end they are embedded or anchored into this protein called villin, and on the opposite end they form in the cytoplasm of the cell the terminal web. So terminal web is the cytoplasm aspect. You can see this nice lattice work here, and then it's embedded here in villin on the tip side there. Now you don't see in this image here, but typically the microvilli are covered by a proteinaceous material called a glycocalyx, which keeps these structures protected from the external environment. Now, when you take a look at stereocilia, which is another structure that I want you to compare and contrast to microvilli. Stereocilia are very similar to microvilli in that they are long finger-like projections. However, these structures have a branching or a bridging configuration. They're located at the apical side of the epithelial surface, and they also contain actin microfilaments. There are 2 areas I want you to know that their locations and their specific functions. They're located in the epididymis with the main role of absorption of fluid. Here's the cell, and then you see here these little fuzzy-like stereocilia. They're very long, they're very fine, and so that is, again, the one location. The second location is in the inner ear, where you have the stereocilia here that are very important for sensory and mechanical reception. Now, when you look at the epididymis here, you have the actin microfilament core. You have these stereocilia are very long fingers, and they're also bridged and branched here. They lack villin and they lack glycocalyx, and what you can also see here is that you can have these presence of these endocytotic vesicles here. The stereocilia in the ear are very unique in that they're organized in bundles of increasing height. It's very unique, very characteristic, and you see there it's very beautiful in a way kind of similar to the rock formations in Utah. What makes this very unique is that the actin microfilaments are produced at the tip, and they're polymerized and cross-linked by a protein called espin. As they are increasing and grow in this direction here, kind of like a treadmill effect, they become degraded and are remodeled in a way. So from here they start off, and then they grow downward, which they will eventually be remodeled and recycled in a way. So on that note, that's basically the brief summary of microvilli and stereocilia. Do know, again, the key concepts that we just discussed earlier. Contact me if you have any questions or need clarifications, and I will see you at the next lecture. Thank you. Bye.